Thank you for inviting me. You've heard inspiring words about gender in science and STEM. And you've also heard about sequencing, which is very relevant to my talk. So what I'm going to do is talk about chromosomes and chromatin and give you a few examples of role models of women who have made major contributions to this field. Actually, this field is one in which many or most of the landmark discoveries were actually made by women. So it all starts with chromosomes. Next slide, please. The chromosomes, human chromosome, as you heard, was sequenced in first complete sequence in 2001. You have 46 chromosomes, 23 pairs, one pair from your parents, or one half of the pairs from each parent. And the chromosomes themselves are made up of DNA and proteins. Next slide, please. And what we know, or let's say the dogma, is that the chromosomes contain the code for life, and each organism has a unique set of chromosomes. So within the DNA, let's say of a human, there are about 30,000 genes. The genes code for different proteins, and those proteins are differentially expressed in different cells to form different cell types. And then the different cell types form the different tissues of all the human being, and working together form a system, which we call life. So as said, this 3 × 10⁹ letters, individual nucleotides, form the human genome. Next slide. There actually is a question: is there really one human genome, and how similar is one to the next? Out of this 3 × 10⁹ base pairs, there are actually about 2 to 3 million differences between any two people, even of the same race, same background. But few of these differences have phenotypic changes perhaps in minor appearances, but not in basic functions of viability. Nonetheless, in our species, based on evolutionary arguments, tracing changes back to the last common ancestor with chimpanzees, we do see that there are 5 mutations stabilized per species per year, which means that even man is en route to evolution. Now, where do these changes come from? There are actually many, many chances for changes or mutations to accumulate in our genomes. And so, in fact, in an adult body, you actually have many different genomes, in a sense, because they're carrying many different mutations. So to make a human body, it takes about 300 billion cell divisions, and there are about 40,000 to 80,000 potential lesions, single nucleotide lesions, per cell per day. So even with a somatic, this is, of course, most of these lesions are repaired, but this gives you a somatic mutation rate of about 10 to the -7th, which still means, next slide, that there's a massive chance for mutation every time a cell divides. Next slide, please. So for each cell, oops, sorry, back. For each of us produces about 50 million new blood cells every day. And with that production of new blood cells duplicating the genome, we have at least 12 × 10⁻13 chances of a mutation every day. So it's clear that our genomes are in flux. Now, despite that flux, that's on the single nucleotide level. If we look at chromosomes, again, the structure, the general karyotype of the human chromosomes, you can actually hybridize different fluorescent probes to them and see if they have altered. And there are dramatic alterations. This is a normal human karyotype where you see 2 of each chromosome. But actually in cancer, particularly in lymphomas, you see quite a different picture. here, using the same chromosome-specific probes, fluorescent probes, you see that there are a number of chromosomes that are mosaics. In fact, they arise from translocations, or their arms, such as on chromosome 15, where you're even missing a single arm of chromosome. So rearrangements and translocations are very common and are very typical marks of cancer cells. And along with translocations, you also get aneuploidy, as shown here in the human, in a carcinoma cell, where actually the cells have fused probably or failed to divide. So you have a multiplicity of chromosomes, among which are many with translocations. So question really is, how do these translocations arise? I mentioned point mutations, but translocations, they arise in a different manner. They arise from what we call double-strand breaks. That's when a chromosome, here in this yellow with the blue caps, when a chromosome actually gets a double-strand break, that means the protective cap that protects the end, called a telomere, is lost. That makes a very recombinogenic end, and the two free ends after replication are very likely to fuse. Now, this fusion, if you don't go through cell division, it's not too toxic because the genome is basically still intact. But in fact, as the two chromatids are pulled to opposite sides into daughter cells, as you see here by the spindle fibers, then the fused chromosome is unable to divide, obviously, because it's attached to the being pulled in both directions. And there's breakage again. And then the two halves, or the two chromosomes separate, but with unequal chromosomal ends. And you have a free end again that is liable to fuse with another free end, and the cycle sets up. It's a persistent cycle of breakage and fusion that allows for transformation of normal cells into cancer cells and to augment the chromosomal defects in cancer cells. Now, the person who discovered this was not studying cancer at all. It was a Nobel Prize winner, Barbara McClintock. She was an American plant geneticist who was studying corn or maize and was looking at mitosis and looking at chromosomes for many years, trying to understand what caused different phenotypes in corn. She observed this cycle of fusion and breakage and noticed that the breaks in chromosome arms often occurred at sites, repetitive sites. And she was awarded the Nobel Prize actually when she was 81 years old. She was hardly recognized actually for the great geneticist that she was. Now, what she got the Nobel Prize for was not for this cycle of fusion and breakage, which is actually crucial for understanding genome rearrangements during cancer. She was awarded the Nobel Prize for yet another discovery called transposable elements. And in studying the maize, these are kernels of corn that you see, and looking at what caused different colors of corn kernels, she identified a gene, let's call it C, that causes dark colored kernels, and noticed that when it was mutated, and that mutation could arise by insertion of an element into that gene, then the dark colored kernel became clear. This would be a gene disruption. But what she noticed was that these inserted elements that can disrupt genes can also be mobilized by a second element that she called an activator. So this DS is called, is a dissociation element, and the AC is an activator element. And that led to these kind of speckled or randomly colored kernels, where when it was the DS element was popped out, the gene was restored. In some cases, it'd be dark colored. In other cases, it was left mutated and it would be dark. So this kind of random, spontaneous mutation through an element that she could show was transposable was shocking. It was ignored in the 40s when she discovered this and described it. Finally accepted as a mode, as an integral mode in all genomes, actually. All eukaryotic genomes have these transposable elements in the 1970s, and the prize was actually given to this when she was 81. Now, these transposable elements are, many of them, remnants of viruses that have invaded our genomes and left endogenous elements that can be activated to jump around. So it's obviously very important that we keep these under control. Now, the translocations that often occur at sites where there are transposable elements, and their excision often leaves a double-strand break. Now, another woman hero in the field of chromosomes is Janet Rowley. She did not get a Nobel Prize because she actually died before she could get it, I think. She lived from 1925 to 2013. Quite different from Barbara, who never married, had no family. Janet was an MD, had 4 sons, and worked part-time through much of her career. But what she was a cytogeneticist, and she studied the karyotypes of human chromosomes in many different situations. And what she found was that there were reproducible translocations. They were not random. And she found that in certain cancers, particularly in lymphomas and leukemias, there were reproducible translocations of bits from one chromosome to another chromosome. And she identified in 1973 a translocation from chromosome 9 to chromosome 22 that occurred always in chronic myelogenous leukemia, so CML. And this was called the Philadelphia chromosome. It was later shown that the result of this fusion created a constitutively active receptor tyrosine kinase that drove cell division and therefore was the cause of the cancer. Now, this inspired chemical companies or pharmaceutical companies to develop a targeted inhibitor that would be specific for this fusion kinase, because it would only be found in the cancer cells. And indeed, it led to a therapy called Gleevec, which was a breakthrough therapy, has made billions for the company that sells it, Novartis, and has been considered one of the true cancer cures because it inhibits the oncogenic gene. Now, the transposable elements that are often at the basis of these breaks make up about 70% of our genome, or transposable elements or repeats. So the vast majority of our genome and the vast majority of the Illumina sequences that they have to deal with are actually repetitive DNAs. And only 1 to 1.5% of our genome is actually encoding genes. The rest, a large fraction, about 17%, are introns, and another 16% are non-repetitive but intergenic sequences. But the vast majority is junk. So what is this junk and how do we deal with it? Well, this is the distribution of repeats in the human genome by percentage. And some of the repeats are not encoding transposable elements or viral transposases. Rather, they are just nonsense DNA, like simple hexamers or simple dimers or trimers that are repeated many times. Some of them have been put to use by the chromosome, such as the telomeric repeat, which is a hexamer found at the end of the chromosome in hundreds of copies. It provides a cap for the end of the chromosome. Centromeric repeats or centromeric satellites are repeats of about 170 base pairs, amplified head to head, extending over megabases surrounding the centromere. And then there are random micro and mini satellites scattered along the chromosome arms, which are non-coding, as far as we know, no use, They're simply repetitive DNA. That's passenger DNA, junk DNA that burdens the coding sequence. But the worst of all, or the heaviest load, is actually this vast number of transposable elements. They make up 40 to 50% of our genome. They often carry their own transposases or their reverse transcriptases that allow them to be transcribed and then as external or independent genomic elements to transpose and insert in different sites in the genome. So it's obviously of a major importance to the integrity of our genomes that we keep these transposable elements and the repeats actually packaged up and silenced. Now, before I talk about how that's done, I want to talk a little bit about the useful bits of repeats. And some of those, as I mentioned, are telomeric repeats. So telomeres are hexamers, are made up of hexameric repeats, T2A3G. That's true for almost all species, vertebrate and animal species. And they're found coding at the end of chromosomes, both as a double-strand repeat and then with a 3' overhang. Now, 2 women were awarded the Nobel Prize for characterizing and proving that these repeats were essential for maintenance of the chromosome N because they identified the enzyme that actually allows their replication. And this was Elizabeth Blackburn and Carol Greider. They got the Nobel Prize in 2009. Carol was the student of Elizabeth Blackburn, and then this is one of the rare cases where the student actually got recognition for helping to isolate this enzyme. So what they showed was that there actually is an enzyme that can independently add these repeats to the ends of chromosomes. Now, the reason that's important is because telomeres without this enzyme, so this enzyme is a an RNA protein complex called telomerase, which self-primes and extends the end of the chromosome with a TG-rich repeat. Without this enzyme, telomeres, or the ends of chromosomes, naturally shorten over time through cell division and in the end lead to cell senescence. So when your skin cells or any of our cells are terminally differentiated, they go through a limited number of divisions. And this is due actually to the senescence partially triggered by the shortening of chromosome ends. Now, tumors or cancer cells express telomerase and therefore become immortal. And inhibiting telomerase obviously would be one way to keep tumor cells from proliferating. Now, we can't get rid of telomeric repeats. If we do, then we end up with the recombinogenic end that causes fusions and breakage cycles and drives cancer. So a further discovery that was crucial to understanding how chromosomes function was identifying the protein that binds this repetitive sequence. It's called the telomere repeat factor 1 and 2, that bind these hundreds of repeats of T2A3G and allows it to fold up into a looped structure at the end of the chromosome so that the end of the DNA is not recombinogenic. And demonstrating this was the work of yet another woman scientist, Titia de Lange. She's still professor at Rockefeller. And she showed that these proteins and the repeats are essential for protecting the end of the chromosome, just as Blackburn and Greider showed that there was an enzyme that could extend them. Now, this is highly conserved. And another woman that maybe you know because she was a professor at NTU showed that the mode that this TRF binds DNA, which is shown here, it's two helix coil turned helix coil domains. She showed that these bind the T2A3G repeat and stabilize this telomeric factor on DNA. She also showed that this is conserved. And even in budding yeast, a simple single-cell organism, there's a protein called RAP1 that binds the telomeric structure with the exact same fold and protects the DNA fiber with its binding. And here you see 2 molecules of RAP1 binding the telomeric repeat. Now, what's the importance of that? What happens when factors bind repetitive DNA? I've said that it can protect the end, but are there other implications? And yet another woman scientist made a fabulous discovery that when RAP1 binds the telomeric repeat, it not only protects the end, but it sets up the repression of nearby genes. So if you insert a gene, and this here we've put a marker gene that will turn yeast cells red near the telomere, if the marker gene is off, the cells turn red. And when Ginger Zakian introduced this marker gene near a telomere, she saw that there was a heritable repression, of, sorry, oh, I can't see this. There was a heritable repression of the genes, of the gene near the telomere. And this was variegated. It was stochastic, just like what was observed by in the transposable elements. But in this case, there was no transposition. It was a natural occurrence of a variegated expression. And this important telomere position effect was discovered by Ginger Zakian, who just retired as a professor from Princeton. It gives this kind of patterned, but randomly sectored yeast colonies, where some cells will be on and some cells, the genes will be off. So the question was, how does this work? And actually my lab worked on this. It's a question of how can you silence genes in a heritable manner. And what turns out is that RAP1 binds a protein called SIR4, which then binds another protein called SIR2, which deacetylates histone tails, which are organizing the chromatin in subtelomeric domains. And then a third protein called SIR3, and these SIR proteins all bind each other, binds to the nucleosome and then represses axis for transcriptional machinery. So this propagates along the complex, spreading along the chromosome until it runs out, basically, of the SIR234 protein. Now, this establishment of this is very much dependent on the concentration of proteins, and that is why actually you get a stochastic repression. But this same type of stochastic variegated repression, it's an epigenetic phenomenon, was discovered by Sarah Elgin when she was looking at the position effect variegation in flies. In the fly eye, it happens also to be a red color. If the gene is repressed, the eye color is white. And actually, if you put the white gene near a repetitive sequence, you get the same kind of variegated stochastic repression. This is what we've now identified as a typical phenomenon for epigenetic repression. And epigenetic repression depends very much on the structure of nucleosomes. So I said all this was due to proteins binding next to the repeats, but they're binding actually nucleosomes. And nucleosomes are the core element that organize the fundamental DNA fiber by wrapping it 2 times around. And each nucleosome has 8 histone tails, which actually are the binding sites for these various factors. So nucleosomes, as they organize the chromatin alone, they are repressors, but it's the modification of those little squiggly lines, covalent modifications of acetylation or methylations, that allow the regions to either be open for transcription or closed for repression. And it's the modifications and the binding of these modifications that lead to variegated and or repressed gene expression. So the heterochromatin, or the variegated gene repression that I've shown, is very much in most organisms dependent on this methylation on lysine 9 of histone H3. Now, another woman was key to understanding how chromosomes are organized into nucleosomes. Her name's Carolyn Luger. She's a professor in Colorado, and I'm sure she'll get the Nobel Prize one of these days. She determined the crystal structure of the nucleosome, which is shown here, published in 1997, a total landmark paper showing how the DNA is wrapped around the nucleosome. And then, as you see, these thin blue-green lines sticking out, the histone cells would extend from the nucleosomal fiber, allowing sites for modification and then repression. So what we've learned over time, next slide please, is that the modifications are key to gene regulation, and that these epigenetic marks are conserved. They occur in organisms as simple as yeast and up through to humans. And in my own laboratory, we made mutations in the enzymes that modify these chromosome ends and could show that the methylation of these lysines, particularly of lysine 9, is crucial for the establishment of what we call constitutive heterochromatin that will silence genes that are near repeats. But we also showed that these play a crucial role in silencing repetitive elements. So I'll skip the next slide and go on. What we know is that if we do not silence these elements and set up the heterochromatin, the epigenetic marks that silence, that silence genes and silence repetitive elements, then these transposable elements are de-repressed, as are the simple repetitive DNA. This allows massive genome reorganization because the transposable elements, those which have active transposases, are activated, they're transcribed, they create small viral particles, they reinsert in our genomes, they disrupt genes, and basically cause cell death if they're fully activated. But the transcription of simple repeats is also highly deleterious and leads to premature aging due to actually the creation of blocks to replication. So it's crucial that we keep these transposable elements silent. It's crucial that all these steps in genome control, chromosome organization, and chromatin organization, all discovered by women, all mechanisms that I described today are the fruit of women, that they're essential for the propagation of our species and understanding the protection of our genome. Now, I want to leave you, of course, what happens if they are expressed and repeat sequences are transcribed in cancers, in aging cells. So if you compare the transcriptome of babies and elderly folks, you find that these transposable elements are expressed in aged persons and not in young children. We also see them being expressed in degenerative diseases and in response to stress. So these are real markers of disease and cancer as well. The satellite DNA is expressed in almost all DNA, centromeric satellites, but also other repetitive DNAs are expressed in almost all cancers. 80% of cancers are expressing repeats, while no normal tissues actually express these repeats. They're kept silent by the integrity of the repressive chromatin. Now, what I've told you about is a series of discoveries extending from the 1940s up through last week, practically. I mean, some of these discoveries about keeping repeated elements silent and the effects of disrupting the histone H3K9 methylation were only published last year. And what I hope I've given you is examples of women scientists who have contributed in a fundamental way to understanding an entire field. Now, this is an interesting phenomenon. Why are there so many women in this field? And I think it's a self-propagating, it's almost like heterochromatin, it keeps propagating. But once you have a critical mass of women in a field, they attract other women. They're women as role models. Any of these women would be a worthy role model for you, I can tell you. They're all, most of them I know personally, except the ones who have passed away. And they're fantastic scientists. It's a congenial, idealistic field where science is the most important thing, science and discovery, and not who you are, nor honors. It's a topic that is highly medically relevant. And I have to say that women train other women, just as Carol Greider was trained by Elizabeth Blackburn. It's clear that this is a field where role models have played a major role. But it's not only that. Most of these women actually had men as their mentors. And so what I would say is the important thing is just to believe that you can succeed, to find a field and a topic that you believe in. And really it's the empowerment of women to succeed that is essential to making sure that women pull their 50% load that they should contribute to the discoveries of chromosomes and chromatin and let's say, the advancement of scientific knowledge. So that's all I wanted to say. Good luck with your careers, and hope your faces appear someday. Don't leave it to chance. Take your career in hand, and don't relinquish your dreams. Thank you.